I'm going to talk to you about the perspective from the genetic signature of the environment. It's a bit complicated, but it is what environmental DNA is. So you all know that we are losing biodiversity very, very quickly, and we know that from charismatic species, tigers. We don't remark it on swallows, but we lose a lot of abundance in swallows. And some species like sharks are not very well known, but we know we lose them a lot. And of course, healthy environments are vital to human well-being. They provide welfare to local people, from fisheries to well-being. This is what we call One Health. And now, biodiversity also provides ecosystem services, and that's a lot of species that are not well known to the public. Of course, the bees are, but not the columbola and not the earthworms, which are vital to agriculture. And monitoring biodiversity is challenging. It's challenging because you have challenging environments, the earth, for example, the ground. It costs a lot. And now we lack more and more taxonomic expertise. We lack systematics. We lack people able to recognize a columbola, a bee, or an earthworm. So most of the time, only the charismatic groups are regularly surveyed and monitored, like birds, bats. But what about the 99% others? No one has a look at that. So what about another way to monitor biodiversity, something more efficient, cost-effective, quick and easy, detecting and identifying all life, and standardizable for monitoring on a long-term basis? Would you inventory 600 kilometers of a mega-diverse tropical environment? Let me try. What is environmental DNA? All of us are losing cells, mostly skin cells, like we lose every day 500 million skin cells in the environment, just touching something. A lot of my cells would be there, and those cells have my DNA. We lose 18 kilos of skin cells on our life. It's just a lot of information, as we were talking before, that is on the environment. So basically, the idea is to collect those cells in the environment, in water, for example, and to sequence them, and to match them with the reference database and see who was there. How does it work? So we just pump water and filter it with high-capacity filters. Then the samples are sent to the lab. The DNA is sequenced with NGS sequencing methods. And then we match the sequence fragment we get from the environment to reference databases. And we end up with a list of species per sampling site. And this is infinite on the paper. You can identify whatever you want, every form of life, animals, cells, bacteria, plants, whatever has this DNA information in it, so it should work. And it does. There's more and more paper on environmental DNA, most of them focusing on the method itself. But now we have people trying to implement environmental DNA for answering big biological questions, and this is what we want to do in Spygen. So what we know is that the detection probability is generally higher with environmental DNA, at least for some of the animals, like amphibians, fish, and freshwater bivalves. And this is important, because I was talking about challenging environments. Big rivers are challenging environments. How can you survey that? If you're thinking about this small bivalve species, which is very sensitive to water quality, Odner Pisidium wateserianum, you see the size. How can you detect it in a large river? Diving in troubled waters, in dangerous conditions. You can have navigation, you can have depth. Well, it's very hard to get there, especially if you want to find this small thing. We detect it very well with environmental DNA. We implemented environmental DNA in France to survey those freshwater bivalves, which are very hard to find and very hard to identify. And it just changed the picture of the conservation of some species. Some species were known only on the hatched areas. And in A, you have a threatened species. In B, you have an invasive species. And the gray area is what we can draw with environmental DNA. Some species, on the contrary, were thought to be widespread, but environmental DNA shows that their distribution areas have shrunk a lot in the last 10 years. Going back to my challenging environment, so this is the Maroni River between French Guiana and Suriname. And we decided to survey all life there, from bacteria to large mammals, using environmental DNA. So we took boats, and for a month, we went from the mouth to the spring and made all this sample all the way around. And it worked. It worked. We did a big transect from bank to bank, and we managed to detect almost all what was known in the Maroni River. A few examples, 100% of the known fish species, we detected them, even the small ones living in the small streams. 60% of all the known mammals, including bats, so we detected a lot of bats. One was new to Guyana. 100% of the monkeys don't go in the water. Some of them never go in the water, and we detect them because the eDNA is drawn down in the river, and we detect terrestrial species very well. And new species to science that have to be discovered. The application was here, this publication, on the impact of deforestation on all life, well, here, fish and mammals, where they could show that the impact was much further than the actually impacted area, and that a lot of species are disappearing even 30 kilometers away from the deforested area. Another challenge in environment, for example, is subterranean environment and subterranean waters. So it's very hard to go and sample there. Some people do dive in subterranean waters. I did a bit, but it's really challenging, especially if you want to look at those very small, tiny mollusks that live in subterranean waters. So behind our tap waters, they live in the water we drink. So they can tell a lot of things about that. This is what we could draw in southern France about the water systems that behave just like islands. So investigating the fauna in the subterranean waters, you can draw this map of several islands, and these are several resources of drinking water that are completely isolated one from another. This is what the fauna can tell. And to access this kind of information, of course, you have to know the fauna, and environmental DNA is a great tool for that. So this is what I call the taxonomic signature of the aquifer, which is also genetic signature. So we switch from taxonomy, like finding those snails, identifying them, put a name, to a genetic signature where we don't even have to know who is behind the gene we collected. I mean, it's just a genetic signature. And this gives information that is highly valuable to hydrologists because they can study the pathway of water at one time with coloration and things like that. But what they don't know is what happened in exceptional events, like big floods, the ones that happen every 100 years or every 1,000 years. Our fauna, our subterranean fauna, can witness that. Because if there's an exceptional flood, it will pass from a network to another. And this is how we can draw these big islands, which are hard to draw if you use only coloration, which are one-shot picture of what it is. This is another study on the impact on tropical forest management. You probably have heard about FSC. So we compared FSC versus non-FSC forests, and we found that, for example, mammals are not good bioindicators of the management. Everybody focus on elephants and chimpanzees and big mammals because you can study them with a camera trap, for example. But it's not. We see that here it's not really relevant. What is relevant, or fish are not relevant either. What is relevant is mushrooms, chromists, and frogs. No one studied them. With environmental DNA, we show that these are the groups that make the big difference. So I'm talking about qualitative analysis. comparing number of species from a place to another, but we can also go towards quantitative analysis. So I said mammals there was no difference in mammals between these two modes of forest management. But if you focus on quantitative, which means the quantity of DNA you find in the water per species, and for example, on hunted species, there's a big difference. So it means that you find the same species on both forests, but much more of them in the FSC managed forests. Another example, which is really a bit new, and a lot of people are working on that, but it's a bit hard to find conclusions, is the impact of agriculture practices on the ground. The life in the ground is mostly mushroom, earthworms, bacteria. No one will collect those things and identify them. eDNA can. And we show that there are a lot of differences between the management in the seagull farm, between the vineyard, fallow land, the driveway, the mulch that was spread on the ground, and the hedgerow. It's hard to interpret, but we see that there's something there. And eventually, just to talk a bit about marine, it works very well in the sea too. This study was showing that in a protected marine area, there's a bit less biodiversity because you lack the small fish. You protect the big ones from fisheries, but the big ones eat the smalls, and there's a bit less biodiversity in those protected marine areas. Well, that's it. I finished with a picture of me sampling on a big lake in Africa in a much safer way than if I had to dive between the crocs and the hippos. Thank you.
Thank you, Vincent. As far as I understand, it's like a signature of the biosphere, in fact. So I have a question in terms of, let's say, international undertaking of that. I guess this is also can be used as a sign for climate change because you can monitor that in the change of the environmental DNA. So what are you already talking to regulatory bodies to propose policies to monitor that at a global scale?
This is what we want to do with the VigiLife project. The VigiLife project, that was the project in the Maroni River. The idea is that to take pictures every 5 years and to monitor big changes. And we know, for example, I was supposed to go next month to Guyana to sample again the Maroni River. And we are not going to go there. We wanted to go very upstream where no one lives. Even the Amerindians don't go there. It's a very tough expedition, but it's a very nice place. And we can't go there because it's drought. We can't pass with the boat. So even the most remote places in the world are highly impacted by climate change, and eDNA can show that.
We have time for one question. Willem?
It's maybe an odd one, but there are people who believe in Bigfoot, a mythical creature that lives, I think, in the Pacific Northwest in America. In principle, you should be, at least give absence of their DNA is not proof of absence of the creature, but you should give a kind of probability estimate, like if it had lived there, I would have found their DNA in a sufficiently large sample. Have you ever thought about doing that kind of... I've
been approached by these people.
Oh, you have?
Yes. Yes, well, yeah, we can work on that. I mean, people have been working on the Loch Ness Monster, on the Yeti in the Himalaya. Well, it's nice. It gives us money to work, and to work on something else.
But the other way around, did you measure or collect a DNA that you could not relate to any known species?
Yes.
Because that's maybe another way of looking at the question.
Yes. I was very excited to go on the Upper Maroni with this drought because there are 6 mussel species that are known only with eDNA and never been found.
And
that would have been, but well,
that's a local Loch Ness.